Hi, my name is Madison Eppers. I'm a senior at North Penn High School in Lansdale, Pennsylvania, and I currently take a dual enrollment course with my local community college in biotechnology. So today I'll be talking about gram staining, a technique we most recently learned in class. Gram staining is most commonly used in microbiology and health professions to characterize bacteria into two types, gram-positive and gram-negative. Doctors can then use this information to administer the correct antibiotic to patients to effectively kill the cells that they have. Gram-positive bacteria are categorized by a thick layer of peptidoglycan. Most cells with a cell wall, like plant cells, have a cell wall made of cellulose, but bacterial cells have a cell wall made of peptidoglycan. Gram-positive cells are simple cells. They have a plasma membrane and this layer of peptidoglycan, so they require different antibiotics than gram-negative cells. Gram-negative bacteria are more complex, though they have a thinner layer of peptidoglycan. They do have an outer membrane, which makes them more complex and harder to destroy, so they require different antibiotics than gram-positive cells, which are much simpler. When it comes to gram staining, my friends and I like to use this mnemonic device. Come inside and stain. In order to properly stain your slide, first add crystal violet for 45 seconds. It's a purple dye that will fix to cells with thick layers of peptidoglycan, like gram-positive cells. Next, add iodine for 30 seconds. This fixes the dye to the gram-positive cells. After that, add alcohol for 10 seconds. This acts as a decolorizer for cells with thin layers of peptidoglycan, like gram-negative cells, so that they're clear after this wash. You don't want to add this alcohol for more than 10 seconds because then it'll start to degrade the cell. After that, in your final step, add safranin for 45 seconds. This will dye the gram-negative cells that were made clear by the alcohol pink, so that you can see them under a microscope and observe their shape. Recently, my class did a lab where we went to different parts of our school and swabbed for bacteria. This is a picture of my plate of LB agar after I swabbed 3 different areas of the school. After gram staining my bacteria on a slide, these are my results. They're small, pill-shaped bacteria, and they're purple, which means that they're gram-positive rods. Even though this technique is widely used by professionals, it can still be used to find bacteria in the most mundane of places. For example, I found my gram-positive rod bacteria in the girls' bathroom at my school. Gross. Thank you for watching. I hope you learned a little bit about gram staining.